Wonderful. I can see that some of our colleagues are still joining, but I would like to welcome you all to the Pharmacometrics Uganda seminar. And if you've joined the seminars before, you'll know that what we try to do is we try to invite somebody who is an expert on a particular area or many areas of modeling and ask them to present some detailed analysis or a specific case that might be relevant to what we're doing. And then at the end, we allow some time for questions and answers. And so with this Zoom webinar, you'll see a couple of features. You'll see that there is a Q&A button, which is better for asking questions because it makes it a lot easier to moderate them, because what we can do is we can respond to specific questions and you can bump people's questions up and things like that. There is also a chat function that you can use, but if you can remember, use the Q&A. You can also raise your hand, and at the end we will, if you want to speak out your question, that's also absolutely fine. So today I have the delight in welcoming Professor Sandra Cvijic from the University of Belgrade in Serbia. And she was introduced to our team by Henry and Zama, who many of us know, particularly to talk on this topic of mechanistic absorption modeling. And I can see from those of you that have joined, I know that some of you are particularly going to be interested in how she approaches this, because it might well have relevance to some of the challenges that you're currently facing. Sandra has a very distinguished CV. She did a lot of work in Germany and has published more than 150 papers. So she really has got a lot of strong background, which I'm sure she'll share as she goes through. So I will stop talking, except also to say, if you have colleagues and you know that they wanted to join, you can also ping them, but the recording will be available on our Pharmacometrics Africa website afterwards. And I know that sometimes it's really helpful to go back and listen again to the talk, but also I know that with time zones and commitments, sometimes people can't join live. Now, thank you very much for joining us, Sandra. Over to you.
Thank you, Catriona, for the introduction, and I'm really glad to receive this invitation to present to the organization in Africa. I'm also glad to see that the interest in PBPK modeling is spreading all over the world, so it's really nice to see so many researchers in almost every country are interested in the topic. As Catriona said, I have some relatively long background in PBPK modeling. Although I'm a professor, I will share the slides as I speak. Just a second. Yeah, I hope you can see it.
I can see the slides. You're not yet on presenter view.
Just a second. Now.
Perfect. Away you go.
Okay, so as I said, I'm a professor at the Faculty of Pharmacy, but my research is mostly related to biopharmaceutical drug characterization, which includes PBPK modeling, and I've been working in this field for more than 15 years now. So yeah, I used to have, and I'm still having a lot of struggles with modeling. I hope that that my talk will be useful and also helpful to some of you. So the topic for today is related to the absorption modeling and mechanistic interpretation of drug absorption pattern, although I'm sure that you're all aware that physiologically based modeling can also be used to interpret drug distribution and also some other processes. I figured out that you have different backgrounds, so I'll just start with the basic scientific principles, just to introduce you with the subject for the ones who are not so familiar with PBPK modeling. And then I'll also show you some of my own examples, and conclude with some, let's say, just some end comments. physiologically based modeling is one of the tools that's used to assess drug and dosage form bioperformance. It can be used to analyze drug liberation from a dosage form, absorption, distribution, metabolism, expression, and all of the processes a drug undergoes in the body actually depends upon three groups of the factors. It's either related to the drug properties or to the dosage form properties or to the characteristic of the target organism. And we can use different tools to assess those biopharmaceutical properties, as we call them. Something that's routine, is to go into the clinical trials, which can be rather expensive, but they are necessary sometimes. And then in order to assure that clinical studies will go well, we are performing a lot of experimental studies. And nowadays we have these sophisticated, let's say, in silico or computer-based tools. You probably encountered different names. We're using different terminology. I like sometimes to call them physiologically based models, although they're usually referred as PBPK models, but nowadays we also have something called PBBM modeling, so it's slightly different. And in order to understand the differences, I here extracted some definitions from the regulatory documents, just to see what's the difference between them, although they can sometimes be used for the same thing. I think that the widest term here is maybe model-informed drug development, and PBPK is just one type of model-informed tools, let's say something like that. And PBBM is then again another subtype of PBPK. So there are slight differences between them, and you can understand them when you read the definition, but sometimes we can use different terminology for the same thing actually. All of those models, we are calling them physiologically based, they are different than some other tools, also computer-based, used in pharmacokinetics, because they have let's say, physiologically background. These software tools are imitating human or animal physiology, and not just, let's say, representative physiology of a human adult. It can also be a target physiology of a disease group or of patients of certain age, and so on. So this is the reason why these models are called physiologically based, meaning they incorporate a lot of parameters that define certain physiology. They are mathematical models because they are using a series of equations, usually differential equations, to simulate the processes a drug undergoes in the body. It's not really that difficult to explain all of the processes that the drug undergoes in the body, starting from liberation, then through dissolution, absorption, permeation, and so on, can be described by mathematical equations. So you just need to link all of these equations in order to simulate parallel processes and to somehow assess the effect of multiple factors on drug bioperformance. There are some values that are used in these equations, and they are usually either drug specific or some physiological parameters. And the models are called predictive because they enable simulation of something that's happening in vivo based on the data we are using to feed the model. The first models of this type were actually very simple. They were logical but simple. For example, for oral drug dosing, gastrointestinal tract was presented as a series of compartments. And then there was an equation which defined the drug absorption through the biological membrane. It goes to the plasma, and that's it. This is a simple compartmental absorption and transit model. But nowadays, we really have much more complex models which use a lot of input data, but they are able to define the processes that happens in different organs and tissues. They're connected with blood circulation. And not only that, these models can simulate drug concentration in plasma, they can also predict what will happen in particular tissues and organ, meaning that we may extract the data on drug concentration over time in the target tissues and in the target population. This is something that's beneficial, that's an advantage of the software tools of this type, because a lot of this data cannot be obtained in clinical studies. And if we generate the model in the right way, and if we validate it rightfully, then we really can get a lot of results, much more than we can usually get in a routine clinical study. This is a representation of the physiological model incorporated in GastroPlus software package. This is the software I mostly use. I just want to illustrate you how complex it can be, but not that difficult to understand. As for the gastrointestinal tract, we have 9 regions, starting from the stomach to the ascending colon, and each of these compartments is defined by a lot of parameters like, I don't know, length, dosage form, transit time, volume, pH, and so on. When we administer a drug, actually dosage form, the drug is released and dissolved, and it goes into the enterocyte. So in each of these regions, we can treat the drug as unreleased, undissolved, dissolved, or it goes into the enterocytes. The fraction that's dissolved can be degraded, or it can be metabolized in the gut wall. It depends. All of these processes can be simulated by the so-called advanced compartmental and absorption model. And then a drug can either go enterohepatic circulation, or if not, it goes into the portal vein, and then it goes to the rest of the organism. And now the rest of the organism can be described like a compartmental model. It's an abstract model. We can define up to 3 compartments, and we can use another option to describe the rest of the body as a series of tissues and organs connected by systemic circulation. This other the so-called full PBPK model is much more complex, because each of these organs or tissues is then again described by a bunch of parameters. Although it's more complex, it doesn't necessarily mean that it's better. You really need to carefully decide how you're going to represent the rest of the body, and you need to justify your selection. It mostly depends upon the purpose of the modeling. For example, if we just want to focus on the absorption phase, or we want to assess the influence of formulation parameters or some changes, for example, polymorphic form of a drug on drug absorption, there's really no need to go into the full PBPK model. So it's enough just to use the compartmental model. But if our target is to assess drug concentration in a specific tissue or organ, or if we want to perform simulation for the specific population group, or if we want to switch from one population to the other one, then it's logical and justified to use the full PBPK model. So at the start of your modeling process, you need first to decide which model to use, and again, to repeat, to justify your selection, because both models can be just fine. There are generally nowadays 3, let's say, ready-to-use software packages, which are mostly used by the Researchers Academy and regulatory authorities. It's GastroPlus, or nowadays GPX platform, and SimCip and PKSim. And although I call them ready to use, it doesn't mean that they have all the data integrated. You still need to search for a lot of data. you need to feed the software, but it provides you with mathematical equations. You sometimes need to select and choose, but you have them, and you have data on certain physiologies, which is a lot. On the other hand, someone can generate its own, let's say, in-house model. It can be done in MATLAB, but for something like that, it's not enough to have a background in biopharmaceutics. You need also to have computer skills. So it can be done by a team of people. And there are a lot of examples in the literature where at some universities, people have generated their own in-house models. Myself, I like to use this, let's say, ready to use, because I have background in biopharmaceutics, but not that much in programming skills. So it actually depends on what you want to do, and do you have a team of people, or it's just a couple of you? There are a lot of choices. And another thing to note, these models can be used, and they're usually, I mean, most often used to assess drug bioperformance after peroral dosing, but not necessarily. They can also be used to assess how the drug behaves in the body when it's administered through alternative dosing routes. For example, I have experience also with intraoral dosing route and inhalation. It means pulmonary route. But for each dosing route, you really need to also know a lot about the physiological processes a drug undergoes after the administration. When it comes to the necessary data for the simulation, I've mentioned that some of the softwares are ready to use, meaning they have data regarding the physiology. You can choose them from the software database. But as for the other data about the drug and about the formulation, that's something that you need to input as a researcher. And in my experience, this phase of the collection of data is the lengthiest one, sometimes the hardest one, because when it comes to the data about drug properties, you can find a lot of different values. The software uses quantitative values, so you can find a lot of values that can not just be different, they can even be contradictory, and you can test them experimentally sometimes, but sometimes this is not an option. So in the situation when you are facing lack of data, both experimental and from literature, you can use some other in silico tools to generate the data, like based on the drug structure. They are you can never be certain whether those parameters are correct, but it can be used as a wild guess, better something than nothing. In any way, there are a lot of data regarding the drug that you need to collect or to obtain experimentally to check the data about the physiology, and it really takes time and knowledge. And then there's just one click on the start button and the software is performing simulations, so you can get a lot of data about pharmacokinetic parameters, including numbers, but also graphical illustrations, which can be quite useful. Some more details about the input parameters. This is just an illustration about a group, let's say, of parameters that can influence drug performance after oral administration. And you can see that some of the data are drug-specific, some are formulation-specific. So when it comes to the drug-specific data, there are a lot of things you need to take into consideration. It's important whether the drug is a weak base or acid or a neutral compound. Is it lipophilic or hydrophilic? Is it a small molecule which can go through between the cells, or it's a large molecule? Whether it's charged with one or not. Molecular mass is important, so that, yeah, you can not just assess its transport through the membrane, but also diffusivity in aqueous environment. And then you need to know the stability data under different pH conditions in order to determine how the drug will behave and how it will be dissolved and where it will be dissolved in the body. So there are a lot of things to consider. And beside these drug-related properties, you need to think about the physiological parameters, how the pH changes in different compartments, what is the volume of the fluid, what's the composition. For example, in the GI tract, we have bile salts, we have lecithin, they are naturally occurring surfactants. What is the viscosity? When we eat food, depending on the food type, viscosity of gastrointestinal fluids is increasing. That influences drug dissolution. Gastrointestinal motility is important for the transit time and so on. And I'm repeatedly saying we need to consider a lot of parameters. The software, like PBPK model, is able to simulate a lot of processes, but you are the one who is going to tell the software which processes to simulate. So I tend to collect as much data about the drug and formulation and the target physiology as I can, and then draw my own schemes and try to imagine in my head what are the processes that this particular drug can undergo in the body, and then I start to model. So you cannot rely just on the ready-to-use, let's call it that way, software. You need to think about a particular example, about each specific drug and formulation, and as I said, target formulation. Because the physiology changes with, for example, aging, or the conditions are also different in different population groups. You need to take this into the consideration as well. These software tools that are, again, let's say ready to use, they can offer some databases for some of the population. For example, GastroPlus has, they call it American, it's not Caucasian, it's a mixture. Anyway, it's one of the population, then two from Asia, Japanese and Chinese. They still don't have African Black population. I've heard that SimSip has a version of that population. I really do hope to see that all of these software tools will have in their database a lot more population, and we can expect something like that, because from time to time they are upgrading their software versions. But yeah, so far for GastroPlus, these are the ethnic groups the software has in its database, and then there is a healthy population and some disease population, and you can change the year and gender, and for females, pregnancy state. But beside these default physiology, a user is also able to define its own physiology. These are just some of the examples. For example, if you want to assess the outcome for the patient suffering from achlorhydria, you can just change pH in the stomach. And for example, if you want to perform simulation for the Indian population and you only have Caucasian database, you can change, this is one of the parameters that usually change, colon transit times shorter. Then you can simulate different surgical procedures like gastric resection, intestine selection, resection, colons resection. You can change hepatic blood flow, kidney blood flow, glomerular filtration rate, and so on. And for intraoral dosage form, with aging, saliva flow rate is changing, diffusivity, and so on. These are all the parameters that you can manually change in the database. The software is offering those data, but you can change them all if it's justified, surely. A few words about the modeling strategy. I mentioned that data collection, in my opinion, is a very serious step, and usually the lengthiest one. But since we often cannot find all the data that we need for modeling, there is often a need for parameter optimization. This is a risky step because when you figure out that a certain input parameter is definitely not logical or it cannot lead you to the expected outcome, it's justified to perform optimization, but you really need to be a kind of certain why you're performing something like that and to justify your approach. Once you collect all the input data, you're ready to perform simulations, but in order to be sure about your results, the model needs to be validated. What does this mean? This usually refers to the comparison between the simulated values with some observed data. In order to do so, you need to have some clinical values like PK parameters or CP time profile or urinary excretion profile, and not just for one dose and for one formulation, because if the simulated values are matching the observed values for a single case, or a single dose or a single dosage form, it can just mean that you had luck. In order to be absolutely sure that the model is predicting accurately, the wisest thing to do is to compare the results with the observed data for different doses, for different dosage forms, and if possible, for different dosing routes. Then you can be sure that the model performs correctly. It's usually not possible to work with different dosing routes. I mean, sometimes it is, but in most of the cases it's not. But at least with different doses and different dosage forms, it's quite helpful. And this is the recommended way how to use a generated model, to first validate the data and then use the model to, I don't know, use a lot of things. It can assess how the changes in formulation or for example, polymorphic form of the drug will influence the outcome, to explain what is the absorption mechanism, for example, to assess the effect of food or concomitant use of alcohol, or to perform simulation for different dosage forms or different population groups to justify biowaiver, and so on. I don't need to read all of this. But what I wanted also to highlight is that sometimes, especially at the beginning or after drug discovery, at the beginning of the formulation development, you don't have any clinical data. And then the software can be used for the so-called early predictions. Those are the one, let's say, before first in human studies. Such predictions are, we have to accept them with a lot of uncertainty. We need to be aware that they may not be the correct ones, but they can still be useful, especially for somebody working in the industry. And one of the strategies to use the software before first in human studies is to design an animal model, because these software tools can also be used for the prediction in certain animal species that are often used in preclinical studies, like rats and dogs and monkeys and so on. So they sometimes use PBPK modeling for to estimate the outcomes in animals and then scale those values to perform human predictions. Again, we need to accept this with great risk, but as I mentioned, it can be useful. And there are, 2 or 3 modeling approaches if we are just using input values about the drug and about the physiology, and if we design our model based on these data and then predict the outcome, it's the bottom-up approach. But we can also start from the clinical data and then use the model to, for example, mechanistically explain why something is happening. For example, you do have a CP time profile. You can deconvolute absorption profile, but you want to find out the reason why something is happening. So we can use the model to mechanistically explain, for example, the absorption process, or to test some hypotheses. I will show you later on in the examples. And there's also the so-called middle out approach. That's something in between. So we are using input data about the drug and about the formulation and physiology. Then we perform predictions, but then we're using clinical data to adjust the input parameters to better match the actual data. And I prefer this middle out approach, I have to say. But it actually depends upon the purpose, which approach you're going to use. Now, in order to show you what I actually meant by this story is to go to the examples. These are some of our publications which dealt with mechanistic interpretation of drug absorption pattern. I will here show you the example on trospium chloride, a lot of other examples. They're all dealing with something different. For example, for the ribamapid case, we assess the effect of mucus binding on drug performance, and for ibuprofen, we assessed the effect of concomitant intake of alcohol on drug performance. This one is a little bit different because we really tried to mechanistically explain some clinical data, so it might be the best example for today. I'll try to be brief. So trospium is a poorly permeable drug. This is a BCS class 3 drug. It has relatively high solubility, so solubility is not the problem, but permeability may be. And this drug has relatively low bioavailability, but what's even worse is that if it's taken with food, bioavailability is decreased by 80%. It's really a huge drop. So we wanted to see what causes such a behavior. And for this reason, we designed a model in GastroPlus. You can see the input data. The numbers are not important here. I just want to say that some of the data were extracted from literature, some were GastroPlus default values, and we needed to optimize some of the values. But we also had to find to use some experimental data. For example, I've mentioned that this drug is poorly permeable, but it also has segmental dependent permeability. We know that from literature. So one of the colleagues in the group, he performed user chamber studies with the rat intestine and actually determined the drug permeability to different segments of the intestine. And then we converted these values to the values that refer to the human permeability values. As for the drug transit time in different gastrointestinal compartments, we could not use the default software values because this drug is an anticholinergic agent, meaning that its transport through the gastrointestinal tract is slowed down. We were fortunate to find the quantitative data in the literature, so we adjusted our model to account for the nature of this drug. And another in vitro parameter that we used is dissolution profile, because we wanted to simulate fasted state. We performed the study in simulated intestinal fluid. And these are our results. You can see that the predictions are matching clinical data quite well. So everything's fine. And then we wanted to see what happens when we administer the drug with food. Okay. Viscosity. We checked viscosity of the standard FDA meal and logically found out that the viscosity is largely increased in comparison to the simulated intestinal fluid. And then we designed a series of hydroxypropyl metal cellulose solutions. So we found out that 1.4% of this viscosity increasing agent actually has the same viscosities as the FDA meal. And we used the solution data in simulated intestinal fluids with increased viscosity as the input value. So when we performed simulations, we could see decreased CP time profile, but it doesn't match the in vivo observed way data. There's something else, definitely. Then we analyzed the structure of the drug. This drug is a quaternary ammonium compound, and it was reasonable to assume that it forms ion pairs with bile salts after food intake. Not just after food intake, but in general, in the gastrointestinal tract, those ion pairs are electroneutral. Yeah, they can go through the membrane via passive diffusion. But after the food intake, the concentration of bile salts is very low because bile salts are used to solubilize some lipophilic components of the food, and we may expect that drug permeability will be decreased. We tested our hypothesis experimentally, again using the using chamber, and we found out that drug permeability is indeed decreased. So we decreased this value in the software, and in combination with the slow and incomplete dissolution, we were able to obtain this blue line regarding the simulated profile. It matches the in vivo data quite well. It's not perfect, but this drug shows really large inter-individual variability, and this is a mean profile. So we can say that our simulations are quite good. And in this way, we were able to, let's say, to demonstrate that our hypothesis about the mechanisms governing decreased drug absorption in the fat state are correct. So they are caused by increased luminal viscosity in the fat state and reduced drug permeability. There's also another example I want to show, and it has to do with specific population groups, in particular bariatric patients. We performed a series of studies with different drugs, and I'll just show you briefly one of the examples. I think you're all aware what bariatric procedure is. It's performed in extremely obese patients. So this is one of the solutions for them to have resection of the stomach, and sometimes duodenum and part of the jejunum is also bypassed. There are a lot of physiological changes in these patients, but they still need to drink tablets. So whether the tablets are effective for them or not, this is a big question. And we wanted to be able to provide them some recommendations about which drugs they can take or not. So, for example, a lot of these patients are suffering from different types of arthritis, and they're recommended to take selective COX-2 inhibitors because of the lesser side effects. But depending on the drug properties and considering the physiological changes in these patients, we assume that not all of these drugs may be effective to them. So we have selected 3 model drugs with different properties. As you can see, toricoxib is a weak base, celecoxib and etodolac, they are acids, but etodolac has let's say pH-dependent solubility in physiological range, while celecoxib's solubility is low, but it doesn't change in the physiological range because pK is quite high. And then we created a database for these drugs. So we used a lot of data from literature. Some were experimental, like solubility, pH solubility data. Some were approximated but justified, like particle size. As for the pharmacokinetic data, we here use the middle out approach. We started from the clinical data and use a software integrated model to actually calculate volume of distribution and clearance and so on. And the calculated values were surely compared with literature reported one, just to be sure that we selected the right values. This is the core. We needed to change the physiology. These are the parameters used for healthy subjects, but after bariatric surgery, stomach volume is decreased and the volume of fluid that is taken with the drug. pH of the stomach is largely increased, stomach transit time is decreased, and in some times of the surgery, the enemy is bypassed. We accounted for a lot of changes. And just to show you the results, for example, look what happens with the toric oxide. Its dissolution after surgery is decreased, and this is total percentage of drug absorbed. This is in healthy subjects. Look what happened after OAGB procedure. It's decreased for more than 50%, which is huge, and it's surely reflected in CP time profiles. But if we consider what the situations for the other two drugs, look for the salicylic oxide. Its dissolution, after this procedure, it will be decreased, but eventually the drug will be dissolved and it will be absorbed in distal intestine. So in general, the extent of absorption and bioavailability, they won't be changed much. Something similar is happening with etodolac, but the explanation is a little bit different. This is acidic drug, so its dissolution, it will actually favor after bariatric surgery because pH will be increased and the overall dissolution will be complete. The same applies for the absorption, and bioavailability will not be affected. So based on the simulation results, we were able to give some recommendation to these patients that they should avoid the toric oxide because it really, there is a good chance that this drug will not be effective in these patients, but they can use the etodolac and salicylic oxide instead. These were just some of the examples, but there's a lot more that you can do. For example, in order to give some kind of a review, we performed a text analysis of a PubMed database publication a couple of years ago, and something that we could expect, the number of publication of PBPK modeling. You can see that it has been steadily increased, which this webinar is also a proof that interest in PBPK modeling is increasing. But when it comes to the application field, look at the dosing routes. It's still mostly used for peroral dosing route, but there are examples about alternative dosing routes also. As for the populations, it's really useful to apply PBPK modeling for disease population groups because clinical studies do not provide us with such data mostly, and this is the reason why the largest percentage of the published models actually refers to the disease group. But again, models for healthy population are also useful, depends upon the purpose, and some of the models concern the adult population, some other pediatric population, lesser percentage geriatric, but the number of publication is increasing in this field also. And when it comes to the area of research, we today talk about mechanistic explanation about drug absorption, and a large portion of the publication is really focused on the absorption process. But the PBPK models are also useful to interpret clinical trials, formulation development, and so on. And I left this for the end, but this is not less important. One of the things that promoted the use of PBPK modeling are regulatory guidelines. We now have guidelines issued both by FDA and EMA. And just to note, maybe you're aware of this, but EMA had guideline from 2019 about PBPK modeling, but now they are expanding the guideline on PBBM modeling and quantitative system pharmacology. So they have a concept paper on the development of a new guideline that's open for comments up to the end of May this year. And this guideline is also expected in 2026. And just to conclude with some takeaway notes, I hope that either you know or I convinced you that these models are really useful in different areas of research, but they are not a magic bullet. We're still facing a lot of issues with modeling, especially a lack of data, either input data or data for model verification. And there's actually a lack of well-defined acceptance criteria. I hope that this new guideline will tackle some of these issues. I'm not confident that it will resolve it, but at least we may get some guideline about how to set the acceptance criteria. Again, I have to stress that the models are not magical, but they can be very useful. Although in order to use any kind of modeling approach, you need to have a lot of knowledge about biopharmaceutics, and then they can really be a helpful tool. I think finished with this, and if you have any questions, I'll now check them, and I can try to answer.
Thank you, Sandra. That was absolutely fantastic. Everything that we want from such a webinar. And I found it particularly fascinating because sometimes some of the models we use are just too simplistic. We know that patients have all of this variability and all of these different things going on, particularly when they're sick. And so your approach has just opened my eyes to, yeah, just how much more we could be doing and how much more accurate it could be. I'm noticing already, I don't know if you can see in the Q&A, but we already have several questions, and I would like to encourage colleagues who are online, please do bring your questions. The first one was Jeff, who has asked that in academic or industry settings, can 10% transporter-mediated absorption be neglected, or should it be considered depending on the context?
Well, in my opinion, it actually depends upon whether you can see those non-linear absorption in the dose range this particular drug is administered. For example, if the drug is administered at low doses, this effect of the transporters will not be visible. So the dose response relationship will still be linear. And in such cases, yeah, I think it can be neglected. But if the dose is higher, if you can see dose dependency, which is non-linear, and if it is important, then it's good to include a transporter. So there's not a definite limit. It depends upon the context or a case. But I will first check what are the doses and whether the effect of the transporter is important for that particular dosing range for a certain drug. Is this a
Great, thank you. Jeff, does that answer your question?
Yes, it does.
Okay.
It does. Thank you. I also see two questions from Martin Mutaka. So Martin, I've just altered your settings so you can actually ask your couple of questions.
Thank you,
Hello.
Professor. Can you hear me?
Yes, yes.
Beautifully.
Yeah, I had two questions. Specifically, I would like to know if drug dissolution and release synonymous when building a PBPK model. For example, if you're inputting dissolution data, does inputting dissolution data mean that that is specific and to drug release? And or are they different and can be modeled mechanistically different? The second question is for controlled release medication, like a slow release or delayed release, that are triggered by specific change in environment like pH. How is that modeled, or are there specific models in the current software we have?
Okay, I think I understand both of the question. Let's deal with the first one. As for the synonyms release and dissolved, I'm always correcting, for example, my PhD students that this is not the same thing. It can be, but it doesn't have to be, because the drug can dissolve. For example, if you have a matrix controlled release formulation, the drug can go through the diffusion, so it's dissolved, and then release is the same as dissolved. But you have, if for example, erosion is one of the mechanisms that dosage form disintegration, then the drug can be liberated or released as a particle, and then it dissolves in the body fluids or in the medium. So now I know that in GastroPlus you have two different options. It's called CRU and CL dosage forms, so you can choose between the two of them. And the difference is because that one treats a dosage form like matrix type, the drug goes in a dissolved form, and the other selection, the other choice means that the drug is released as a particle, and then it dissolves in the body fluid. So it's a little bit slower process, let's say, something like that. So it can be the same, it doesn't have to be. And in GastroPlus, you have the options to choose. The problem is when there is a combined mechanism, when you have both diffusion and for example, erosion. And in such situations, I tend to just test the formulation. I mean, perform actual dissolution studies and just input my dissolution as the input parameter. So not to, one of the choices is to let the software calculate or predict drug dissolution, and the other is to input your own dissolution. But this connects me with your other question. What if dissolution is pH dependent or temperature dependent? If it's pH dependent, then it's good to test drug dissolution under pH changing conditions. It's possible under certain setups, like in reciprocating cylinder or flow-through apparatus or multiple stage pedal or basket. I don't know how familiar you're with the dissolution test, but this is something that can be performed experimentally. And then this can be input in the software. Or if you just want the software to predict the solution, pH is already set to be changed between different compartments. So the software actually takes this into account. And you can try different virtual scenarios regarding drug dissolution. What happens if the drug dissolves, for example, after it transits through stomach and when it reaches duodenum? Or what happens if the drug releases 100% in the colon and so on? You can input different dissolution virtual scenario and just test whether it will have effect on the outcome or not. As for the effect of the temperature, I'm not really sure what you meant by that, because usually in the gastrointestinal tract temperature is around 37 degrees. So did you mean something else? One is
No, I didn't mean something else. I just meant like for their formulations, for example, that for a drug to be released, there needs to be maybe a change in temperature. I was just giving an example, but I think
okay.
it refers more to pH. For example, if it's a control, it's a coated medication, and the coating has to dissolve in a basic pH, not in acidic pH. So I was wondering how that is modeled, or
I see.
yeah.
Well, usually by inputting dissolution data, experimental or testing virtual scenarios, because you have data in the software, how much time the drug stays in each of the compartment. So if you want to test, for example, that it releases the drug in the colon, like colon delivery system, you just, I don't know, do the math and then set up the dissolution to release the drug after, for example, 3 hours. So you will actually see the effect. It can either be virtual or experimental profile that's input. And for gastroretentive dosage form, you also have the option in the software to select. So it will automatically account that there is no dissolution in the stomach and that the drug will be released after it reaches the duodenum. Okay.
Thank you.
Okay.
Thank you. I'm aware we have only got about 2 minutes left, but
Sorry.
No, this is great. Because your questions are fantastic, and I note that your email is on the
Yes.
slide. I have an anonymous attendee who's saying that using GastroPlus, collecting the data related to absorption is difficult. So do you actually have to do in vitro experiments to have data related to the absorption, particularly related to using GastroPlus?
It depends upon, it's good if you're working in the industry, I can obtain all of those data, but for example, for me in the academy, no, I can perform, I don't know, permeability studies, the top, it's a pH stability, PK, log P, and so on. As for the pharmacokinetic data, I usually use middle out approach, so I use clinical data. And then there is a software tool to extract volume of distribution and clearance. And then I always compare the calculated values with the reported values to just be sure.
So what if, okay, so then it brings in Rashida's question, but I'll just sneak this one in. If you don't have clinical data, then how would you validate your model?
If you don't have clinical data, there is no way to validate the model. You can just validate some of the calculated parameters, like you can have clearance obtained in hepatocyte culture, and you can find these data published. So you can check whether the input value is okay, but you cannot check whether the outcome is okay.
Fantastic. I'm afraid I think we probably won't get on to Safa's question about nanoparticles, because I think it will be quite...
Okay, I can just give you a hint that it depends upon the dosing form. There are some specific, let's say, phenomena. Nanoparticles goes after oral administration, like trapping between the villi in the intestine that can be simulated. And for some other dosing routes, you need to improvise because the model is not that sophisticated to now assess the performance of nanoparticles, but you can improvise and perform some simulations. But if you're interested into that, you can send the questions to my email.
Thank you very much, Sandra. And as is often the case, the discussion really does get going. But I want to thank you for making the time to speak to us. And thank you to all of you who have joined. As Sandra says, you can email any additional questions. But it's also great to meet you. And it's also great to meet another passionate modeler in our network. So thank you again for your time.
Thank you for the invitation, and I hope it was useful. So until the next meeting or something like that. Stay well. Bye.
Bye now.